Information is liberating. Education is the premise of progress in every society, in every family. A very good evening to one and all present here, and welcome to our Medicine Forum. I'm Ritula from Mada Medical College, final year, here to take you through this event as your host. And for the ones who are here new, let me give a quick introduction on our Medicine Forum. Medusane is a platform that is rapidly growing to become a trusted educational partner for every medical student across India. Our mission is very simple: to provide accessible, high quality educational resources that empower students to excel in their academic and professional journeys. And we have a vibrant community of over 23,000 YouTube subscribers, and we are constantly evolving to meet the needs of every student. Whether it is for you to prepare for your competitive exams or seeking to build a strong foundation in medicine practices, our team stands out and is committed to mentorship. Medicine provides even one-on-one support and practical insights that goes beyond textbooks and helping them build the confidence and clarity onto their own career paths by providing them on their daily guidance. It is an immense pleasure to be a part of this community, and thank you so much, sir, for providing me with this opportunity. As we approach as we approach World Diabetes Day, we are reminded of the importance of awareness, education, and collective efforts in combating one of the world's pressing health challenges, that is diabetes. In order to have a deeper understanding of diabetes from its physiological and chemical to its management and complications, we have arranged a special lecture series. World Diabetes Day is more than a date on the calendar. It's a global call to action. Diabetes affects millions of lives, impacting not only patients but also families and communities. Through this series, our goal is to bring together experts in the field to share valuable insights that will empower healthcare professions, students, and the broader community to tackle diabetes if actively, even in their daily practices. Each session is designed to address crucial topics for the basis of insulin functions to advancement management strategies, along with the latest guidelines as well. Through this session, we will help you to gain a practical knowledge and skills that are essential for improving your patient's care. We believe that raising awareness and fostering understanding of diabetes is very essential in reduction of the burden on this society. We are grateful to our distinguished speakers and enthusiastic participants for being a part of this mission. Together, let's make this World Diabetes Day a step forward in our journey towards a healthier, diabetes aware world. So, we are on session 5, and the topic for today's discussion will be Unveiling the Molecular Mechanism Behind Diabetes Complications by Dr. Desigamani, PhD, and Professor of Department of Biochemistry of Mada Medical College and Chennai. It is an immense pleasure to have you with us on board today, sir. Now, I kindly request Dr. Raghunandan, founder of Medusane, to deliver his welcome address. On to you, sir.
Unmute.
Yeah, sorry, sir. And thank you, Mridula, for being the host today and kindly introducing such a big initiative of medicine. I thank all the people, those who are behind the screen for arranging this wonderful sessions. As we are on the day 5 of the World Diabetes Day series, we have just gone through all the physiology as well as the pathophysiology of diabetic mellitus, then the complications, then the treatment management. Today, it's going to be a very important session as you are going to dwell into the complications and its biochemical, biomolecular mechanisms. And I think this topic will not be justice unless I call Dr. Desiga Mani sir for as a speaker of today's session. Sir was my professor for biochemistry, and I can wholeheartedly say that the confidence and the knowledge that he builds upon to his students on any topic in medicine is very crucial for not just passing the exams but for the management of the diseases or being any specialty. It's very important to understand the concepts, the roots of the diseases. And I think today the diabetic mellitus roots, we are going to dwell into all the biochemical biochemistry as well as a molecular approach to diabetic mellitus and its complication. And sir has published various publications in the national and international journals, and he is being in the field of medicine for the past 15 years, guiding UGs and PGs under his department. And today it's going to be really a great session for us and also for the medicine community to have him in our channel to discuss about the molecular mechanism behind the diabetic complication. Thank you once again, sir, and the session is yours now.
Raghu, I'm not able to see the slide.
Yeah, but sir, slide is visible, sir.
Okay. I am not able to see.
You can just get back to the Zoom and reshare if you
okay.
can.
Now.
Yes, sir. Now it's moving.
Okay. Can I start?
Yes, sir. Please.
Okay. My sincere thanks to Medusane for giving me the platform to share the acquired knowledge on unveiling the molecular mechanism behind the diabetic complications. First of all, why do we need to identify diabetes? There's a reason for it. Approximately 5 to 7 years gap between the onset and the diagnosis of type 2 diabetes mellitus. It poses a lot of problem. At the same time, at the time of diagnosis, 25 to 30% of the patients with diabetes have already started the complications. And they lost almost 25 to 30% of the people with the diabetes in the US, and it's growing every day. That's the concern. We need to diagnose at the early to intervene the disease. So for today's session, I have 4 objectives to deal. Number 1, complications. What are the complications and associated with diabetes mellitus, and explain why these complications occurs. Similarly, explain the unifying mechanism of hyperglycemia that induces cellular damage. Third, explain how advanced glycated end product induces vascular pathology. Finally, are there any way to stave off the biochemical dysfunction and endothelial cells in hyperglycemia? To start with the first objectives, the number of listed complications: diabetic retinopathy, which is the leading cause for the blindness in the world working age. Diabetic nephropathy, it's a leading cause for the end-stage renal disease. Also, there is diabetic neuropathy, a leading cause for non-traumatic lower extremity amputations, cardiovascular disease and stroke, 2 to 4-fold of increase in cardiovascular mortality and stroke in case of diabetes. These are the major complications associated with diabetes mellitus. As I stated in the objectives, the general features how hyperglycemia induces tissue damage. The general features how hyperglycemia induces tissue damage. Hyperglycemia leads to cumulative long-term changes in the stable macromolecules in all the cells. Also, there is a repeat acute changes in cellular metabolism, okay, leading to diabetic tissue damage. What do I mean by long-term complications on stable macromolecules? We all know the cells is made up of carbohydrate, proteins, and lipids. These are the macromolecule which makes all the cells, And in case of hyperglycemia, there is going to be a changes in these molecule which poses threat to death of the cell. Similarly, repeated acute changes in cellular metabolism. As and when the glycemic control goes very high, hyperglycemia starts, there are multiple abnormal pathways are operating in the cell, which is a leading concern for tissue damage. Also, if the same person has an independent accelerating factors like hypertension or hyperlipidemia or some genetic determinant which are susceptible to damage or increases in hyperglycemia, finally contributes tissue damage. Okay. If you are wondering, the damage occurs into more particularly to subset of cells. Okay. The cells which are in capillary endothelium of retina, mesangial cells in the glomerulus, neurons, and Schwann cells in the peripheral nerve. Why does damage occur only in these cells? If you are wondering, what could be the reason? These cells are not able to reduce the transport of glucose inside the cell. That's the curse. These cells are not able to reduce the transport of glucose inside the cell. So glucose transport rate doesn't decline rapidly as a result of hyperglycemia, leading to high glucose inside the cell. Okay. So there is a free entry of glucose into these cells. That is what poses victimization for these cells. Okay. What are the mechanisms going on inside these cells to explain the complications? I have listed 5. There are many pathways, at least to cover these major pathways. Okay. The first one, polyol pathway. Second, intracellular production of AGE, advanced glycated end product and its precursors. Third and the most important pathway which is getting activated is protein kinase C activation and major enzyme activations. We will see in greater detail each one. The fourth mechanism by which the person gets into complication is increased exosomin pathways activity. And the final unified mechanism through the hyperglycemia-induced mitochondrial superoxide production. Okay, we will deal one by one. First, to start with polyol pathway. Polyol pathway. As a person is having an hyperglycemia, okay, more of glucose, that is approximately 30% of the total glucose is taken into this pathway. For a normal individual like you and me, only 3% of glucose is taken into this pathway. Usually this happens, but in a very milder manner, only 3% of the glucose is taken into polyol pathway. Basically, this pathway is very simple. There is two-step process. Glucose is converted to fructose, intermediary as sorbitol. Okay, the enzyme involved is aldose reductase. Aldose reductase, and the major cofactor which is involved is NADPH. So what you have to understand, in this process, NADPH is involved and enzyme also is involved. So in case of hyperglycemia, 30% of glucose is taken into this pathway. So there is a lot of consumption of NADPH, loss of utilization of NADPH, which poses a threat to the cell, decreases the glutathione, reduced glutathione, okay, eventually contributes reactive oxygen species. More of toxic chemicals are produced in this pathway. And the second molecular mechanism which explains the complications in the mentioned 3 cell types, also in all cell types. Okay, one by one, I'll go slowly. Modification of intracellular protein. Modification of intracellular protein. As and when there is an hyperglycemia, the glucose enters into all the cells, okay, and then it is taken into abnormal pathway to produce AGE, advanced glycated end product. Please understand, it's very simple. Intracellular proteins are there. In case of RBC, there is a hemoglobin. In extracellular, there is an albumin. Intracellular, there are a lot of enzymes and other proteins. Okay. So these proteins undergoes non-enzymatically with glucose interaction. So basically glucose attaches to the protein wherever there is an amino group, extra amino group, non-enzymatically couples with the glucose, and that is what is called glycations. Okay, intracellular protein undergoes glycations. Also, you can see there is an extracellular matrix like integrins. It's a protein which is also undergoing the similar sort of modifications, that is glycations. So what's the problem? When the glycated products is liberated from the cell, okay, it is producing small molecules like age plasma proteins, okay, advanced glycated end product plasma protein. And it's a small peptide which are notorious in nature, and then this can go and bind to the receptors on the target cells. Remember one thing, always the glucose excess only in the plasma. Glucose is excess only in the plasma. That plasma glucose, if at all it has to pose threat, it has to pose to the blood itself. That is extracellular protein or intracellular protein or on the capillary proteins. Okay. So the glucose attaches to all the protein that I mentioned in the extracellular or intracellular or on the capillary protein, forms a glycated product, and then these glycated products slowly degraded when their expiry dates are over, and the glycated end products attaches to the endothelial cells or the smooth muscle cells or Schwann cells or retinal epithelial cell, endothelial cells. Okay, there is a receptor called age receptor. This age product and age receptor couples together and then internalized to produce reactive oxygen species, and slowly it releases NF-kappa B, nuclear factor kappa B, which is an activator of many genes, okay, notorious genes like growth factors and cytokines, which eventually causes inflammatory cytokines and growth factor. These are the mechanisms undergoing in case of glycations. The third and the most important pathway, please listen carefully. Whenever anybody has an hyperglycemia, okay, which leads to more of diacylglycerol, okay, through glycolytic pathway, more of diacylglycerol is produced. That's the trigger for protein kinase C. Protein kinase C is an enzyme, which is a very important enzyme, plays a role in maintaining the glucose metabolism. Protein kinase C, there are two forms of isoforms, beta form and delta form isoform. So what you need to understand is when the protein kinase C gets activated, you can see multiple effects on the cell, okay? Reducing the blood flow through reduction in eNOS, endothelial nitric oxide synthase, okay? PKC, when it is getting activated, which leads to reduction in endothelial nitric oxide synthase as well as ET1, okay, which together reduces the flow of blood and then which may explain the abnormalities. Similarly, when a PKC is activated, you can see there is an incremental increase in vascular endothelial growth factor. As you know, vascular endothelial growth factor increases, there is a permeability, which is also called leaky blood vessels, okay, and that is also poses a threat. Similarly, when you have an increased activation of PKC, you can see there is an increased collagen and fibrinogen, which poses a threat to occlusion of capillaries. Also, people have observed plasminogen activator inhibitor is increased in the case of incremental in PKC activation, which leads to decrease in fibrinolysis and vascular occlusions is a result. As I said in the previous slide, here also PKC activation leads to NF-kappa B. NF-kappa B is a transcription factor for many inflammatory genes, which gets overexpressed, and then inflammation is the result in the case of hyperglycemia. And the last and the most devastating mechanism, when there is an hyperglycemia, leads to PKC activation, protein kinase C, which eventually increases NADPH oxidase enzyme, and then which alters the cellular oxidative phosphorylation pathway, which produces increased reactive oxygen species. In other words, free radical formation, which has a multiple effects we will see in the next slide. Okay, if you got confused, one thing you can remember, the things that are good for the normal functions are decreased and the things that are bad are increased. At least you can understand. Slowly you can go through what are the complications when hyperglycemia, how it actually happens at a molecular level. Okay, the fourth pathway by which we can understand how an hyperglycemia causes at a cellular level complications. Increased exosomes pathway activity. As we know, the person is having an hyperglycemia, which enters into the already mentioned three important cell type. The glucose goes into a glycolytic pathway. A fructose 6 phosphate is produced. Eventually, it is converted to glucosamine 6 phosphate with the help of glutamine fructose 6 phosphate aminotransferase. Okay, glucosamine 6 phosphate. Slowly, it is converted into UDP N-acetyl glucosamine. UDP N-acetyl glucosamine. So this compound gets attached to many protein as well as to many genes and then alters those molecule, causing a lot of threat. Also, the recent findings, this pathway, that is increased exosomes pathway, explains about the insulin resistance. How? Because of this UDP N-acetyl glucosamine attaching to many proteins, many enzymes, and many particular genes, which eventually causes insulin resistance. Okay. More particularly, I put some 2 or 3 genes which are already known to them. Plasminogen activator 1 and then TGF beta. These are the gene gets upregulated and then you can see the phenotypic at the cellular level. Okay, the last pathway by which we can understand complication at a cellular level, hyperglycemia induced production of superoxide by mitochondrial electron transport chain. Okay, if there is an hyperglycemia which poses a superoxide production at all the cells, and the superoxide is very notorious, which is going to be a leading cause for DNA damage, activates the PARP and modifies the uh, uh, glyceraldehyde 3-phosphate dehydrogenase enzyme, which is a major enzyme in normal glycolytic pathway. I'll explain slowly. Please understand. In the previous slide, we learned that reactive oxygen species are produced in case of hyperglycemia. These reactive oxygen species nothing but a free radical. Free radical, you would have learned about it is a damaging factor for all the DNA and other molecules also. Okay, more particularly, there is one enzyme which is affected here, PARP, and that is the leading cause for alterations in glyceraldehyde 3-phosphate dehydrogenase. What happens when glyceraldehyde 3-phosphate dehydrogenase gets damaged? So the normal process of glycolysis is not happening because of the decreased activity of glyceraldehyde 3-phosphate dehydrogenase. Okay, opposes into abnormal pathway like what we have seen in the previous slides, increased polyol, increased advanced glycated end product, and increased PKC pathway, and increases in exosomes pathway. Okay, these are the pathway which can explain us how at a cellular level, hyperglycemia poses a threat to all the cell types. That is fine, but how does the unifying mechanism, previously what we have seen, how it causes macrovascular and microvascular diseases? Okay, microvascular changes. See here, hyperglycemia in the case of diabetes mellitus, we have learned about the mitochondrial reactive oxygen species, as well as there is an abnormal PKC pathway activations and glycated end product formations, polyol pathway, exosomes pathway, which is all a leading cause for diabetic micro and macrovascular angiopathy. Okay, I'll explain slowly. Remember one thing. Glucose in the case of hyperglycemia, very high in plasma. As I already told you, this hyperglycemia, that is an excess of glucose, okay, gets attached non-enzymatically to the protein which are present in the plasma as well as into the blood vessels. Okay, let's see what are the protein present. Major protein which is albumin. Also, there is a molecule called LDL which is also having a protein. Okay, albumin also gets attacked by glucose. The LDL, which has a protein, is also being attached with the glucose. Also, immunoglobulin. I've just mentioned only three. All proteins invariably non-enzymatically attached with the glucose. That is what we call glycations. Okay, glycation, non-enzymatic glycations. Okay, at the end of the day, when the proteins are getting degraded, they produce a small molecules attached to that glucose. Those are called AGE, advanced glycated end products. Advanced glycated end products. Okay, remember these are seen in the plasma of the blood vessels. Okay, when these AGE, advanced glycated end products, goes into contact with endothelial lining of the cells in the capillaries, okay, they can attack or attach to the collagen fibers down the basement membrane, okay, and then causing the protein accumulation, also thickening of the basement membrane. Similarly, when LDL is also affected by the high glucose, trapping of the LDL in the artery walls, also there is oxidation of LDL, which is also being migrated to the blood vessels. Okay. Similarly, as I mentioned, the IgE, trapping of IgE leading to the complement activations and finally to proceed inflammation. So eventually what you see, there is an alteration to the capillary, okay, alteration to the capillary in the case of hyperglycemia by advanced glycated end product. Advanced glycated end product. So what happens? When your capillaries loses its normal function, there's not going to be a diffusion. There's not going to be a diffusion of, sorry, there's not going to be a diffusion of nutrient to the tissue. Also, there is not going to be a diffusion back into veins, whatever the waste products which are formed by the tissues. Okay, everything is going to be threatful, and that is what explains what happens inside the cell around the capillaries. Okay, if it is a retina, then you can see the blindness because nutrients are not going to go because the capillaries have got damaged. And then whatever metabolism happens, whatever toxins are produced, even that is not going to be excreted out. Same thing you can imagine if it is a nerve tissues, there is going to be a neuropathy. If it is a renal tissue, there is going to be a nephropathy. Okay. Advanced glycated end products are metabolized into small peptide, as I already told you, that is increased in circulating as a age, high molecular, low molecular weight. Okay. Simply understand diabetes produces increased input of these such molecules. Diabetes produces lot of input such molecules. What is that molecule? Age. So renal or the kidney, which is supposed to filter it out, unfortunately, even the capillaries in the renal tissues are also got damaged. Hence, it is not going to be filtered out. And that is again causing a threat to the renal blood vessels. And that is what explains a lot of problems in the kidney. Okay, one such simple explanation. As you can see, there is a lot of soluble age. Age is nothing but advanced glycated end product. When it meets the receptors on the mesangial cells, and then there is a lot of activations inside the mesangial cell, which eventually produces PDGF, type 4 collagen, lamin, and HSPG, which increases mesangial expansion, which is the leading cause or the explanation for glomerulosclerosis. Okay, basically there is thickening, there's not going to be a filtration. That is how GFR is reduced, and that is what we call end stage renal disease. It's an advanced stage, slowly progresses into end stage disease. Okay, I've explained about the molecular mechanism, how an hyperglycemia is causing with a various altered biochemical mechanism by which all the tissues are getting damaged. or the tissues are necrotized or died off. Okay. Can we stave off the biochemical dysfunction in endothelial cells induced by hyperglycemia? That could be the most pressing questions. Okay. Right now, there are a lot of compounds available for controlling the diabetes with various mechanisms, but still people are having a lot of complications. Are there any newer research in these areas where we understood there are multiple mechanisms which explains the cell death, or in other words, a lot of complications? Okay. Can we have any possibilities to save off the biochemical dysfunction in endothelial cells induced by hyperglycemia? As we all know, we have to take care of our diet. Along with the diet, WHO positions on HbA1c, you have to reduce or to have a cutoff limit of 6.5%. That's the recommended point. Okay. Most recently, this is what I wanted to stress. Most recent findings, it's not a recent molecule, but in related to diabetes, it is the most recent findings. Okay. There's a small protein called NFE2 related factor 2, which is also abbreviated as NRF2. NRF2. What is this protein physiological role? This protein is also called as transcription factor. Okay. You should know what is transcription factor. Transcription factors are the one which helps the transcription of a particular gene. But this particular transcription factor is useful for many gene transcriptions. Those many genes are involved in detoxification process and antioxidant enzyme production. Basically, this particular transcription factor favors detoxification process as well as helps to fight against the free radicals that are generated. If you would have watched me carefully about the molecular mechanism, which almost poses a free radical mechanism. So that's the reason researchers have found this particular molecule can be a target, okay, NRF2. Basically, NRF2 is a protein which initiates the production of number of enzymes that protect the cell from potentially damaging chemicals that are ROS, reactive oxygen species. Okay. If you would have watched carefully, again, I'm repeating, hyperglycemia poses reactive oxygen species, non-enzymatically and enzymatically with various mechanisms. Non-enzymatically means through glycated end product. Enzymatically, there are various pathways that I have explained, produces reactive oxygen species. Okay. What happens? This particular protein, a transcription factor, which is going to be acting on many genes, more particularly on the gene, there is a particular element called antioxidant responsive element, okay, leading to the coordinated upregulation of ARE-driven detoxification process, and antioxidant genes are upregulated. Okay. If you're asking me any findings so far, the last paragraph you can see, researchers have incubated cells taken from the lining of the human small blood vessel cells with two different concentration of glucose, okay, low and high concentration. Then they used laboratory method to see what effects of incubation with sulforaphane had on a range of complex metabolic and biochemical pathways. What is the sulforaphane? Sulforaphane, a compound found in broccoli, could prevent the metabolic damage to our blood vessels caused by an high glucose, okay, high glucose, by activating NRF2. NRF2. This is basically a drug, or it's been available as a drug also, but is also being found in normal food like broccoli, okay, which is being used to see in exogenous on the laboratory setup. Okay, they've taken the lining of the endothelial cells and then fed with a lot of glucose and then incubated with the sulforaphane. Okay, sulforaphane, and then they could see a lot of biochemical changes occurring in favor of or alleviating the complications of diabetes mellitus. Okay. They've also looked into a lot of protections against oxidative damage by measuring all these parameters. Okay. Quinone reductase, glutathione synthesis, peroxidase, thioredoxin concentrations, and so on. They've also checked the protection against the glycation. As I was mentioning, glycation is a leading cause which produces eventually age products and all this. But when they are incubated with sulforaphane, there is a protection against the glycation, there is a protection against the metabolic stress, there is protection against the lipogenic stress. Okay. They've also done activation of NRF2 prevents biochemical dysfunctions on the endothelial cells incubated with induced by hyperglycemia. Okay. The take-home message, if you're asking me a question, what interpretation did the researchers draw from these results? Very simple. Researchers have concluded that activation of NRF2 may prevent the biochemical dysfunction of cells that line the inside of the blood vessels caused by high levels of blood sugar. What a blessings. Okay. But if you're wondering, only the broccoli has got this sulforaphane, no. There are a lot of other Indian food, okay? A diet rich in activity of sulforaphane with the tomato and other whatever you're seeing, onion also, there's cabbage, okay, grapes, apple, various orange, okay, green leaves, all said to contain sulforaphane. Okay, there is also another research, okay, emerging therapeutic intervention, a drug called bardoxolone methyl compound. Okay, it's abbreviated as CDDO methyl esters, already in trial. Okay, a synthetic NRF2 activator developed for cancer prevention. Okay, it was there in cancer prevention, but now showing a promise for diabetic vascular complications. They have done a lot of studies. Okay, one studies I could see phase 2 study in diabetic nephropathy, improvement of EGFR from mean baseline about 30.3 ml to 24%, which is very promising. Similarly, improved vascular inflammatory markers. Okay, these are the promising update about a drug, newer drug, which may be helpful. Okay, please consider these are the vegetables which also can stave off diabetic vascular complications. Okay. More than this, what I like the most, Albert Einstein once wrote a letter to his own son saying that life is like riding a bicycle. To keep your balance, you must keep moving. This is very applicable in the case of hyperglycemia. I thank you.
Thank you so much, sir. I think it's really a wonderful discussion. I will tell a evidence-based discussion because everything was justified with like as you take every time in my UG day also. Every question will be having an answer then and there, and I think this session fantastically highlighted not just molecular mechanism, but I think recent advances also covered from A to Z. And these topics, as far as I have listened so far, will be very helpful for a NEET PG aspirant also, because now the current questions which are coming in the per se INI Central Institute exam or NEET PG is more focused upon the recent advances. And I think this session is going to be helpful for all. Thank you, sir. And I think Mritula, if you are there, you can just conclude the session.
Thank you, Raghu.
Yes, sir. Let us give our heartfelt thanks to Dr. Desikamani for conducting such an amazing session. Thank you so much, sir, for sowing the seeds of knowledge within all of us. I'm sure that all of us got a much better picture on the molecular mechanism hidden behind the diabetes. Now, before we end this session, I would like to thank each and every one of you for your active participation and making this event more exciting and successful. And I would like to express my heartfelt gratitude to our most dedicated Dr. Raghunandan Sir, the founder of Medusane, for giving me such an amazing and wonderful opportunity. Thank you, sir. And thank you for the whole Medusane team. Thank you for everyone joining with us today. And with this, we will now end the session.
Thank you, Mridula.
Thank you, sir.
Thank you, sir. End பண்ணிட்டேன். Almost in 45, sir, இன்னைக்கு attendance.
Okay.